So I'm going to talk to you a little bit about our latest efforts in our software components and how we can move the field forward in getting more comprehensive analysis of proteomes. And I'm going to outline this in a number of different ways. So first of all, I'm just going to give you a brief outline of some of the development of the tools that we have, and then tell you how we're democratizing these tools and allowing people to not only use the tools for free, but also provide those tools in an open source manner so that we can hope that the people can also work on those tools at the same time and increase the utility of our tools. And then give you our insights and how we've suddenly seen a very rich area of identifications of biomolecules and how this can now really move the field forward rather than just looking at the naked proteome as we always do. So I've lent this diagram from Ariel Bensimon, who's also here at this meeting, and I've just embellished on it a little bit further. But I think, you know, from the first components here, we quite well understand the technologies that we currently use either in shotgun or targeted mass spectrometry. But as we get into more of this SWATH, and really SWATH is just another avenue of trying to push the technology beyond what it was originally thought to do and get into this process of trying to become very complex these days and get very multiplexed fragmentation profiles to try and increase the amount of information that we can actually get rather than having the mass spectrometer only feed us back information that it can deal with. But I think we do realize that there is a benefit of this technique. There's a lot of problems with the technique in terms of the data analysis, and then we have to develop a number of tools to help this. But the overall goal here is to become very broad in our identifications and also very deep, but at the same time provide these tools as very quantitative as well. And really the future is that we see this as now a comprehensive and sensitive, but more importantly, a reproducible technique as we move forward. And whether or not this technique will stay as it is or even morph into another technique is still yet to be seen. But the issue is that once we get to this component here becoming very comprehensive, we really get to see the whole of the proteome and not just the naked proteome as it is in terms of sequence, but we see all of the modifications that are occurred. And rather than doing this piecemeal identification of modifications the way that we do these days, the way we try and identify them by doing this with a priori information in the first place and then go searching for them, we collect all this information and then use that information to bring back all of the modifications actually there. And so this is the process that we're taking. I'll take you through that. So some of the things that is going on in our group still, we have a number of major programs. So the transproteomic pipeline is a stable of our group of where we continually develop shotgun analysis tools in which we can just take raw mass spectrometry data, do the identifications, understand them at a statistical validation level, and also provide these as an open source and a freely available tools. A lot of this feeds into what we call the Peptide Atlas compendium, which is an umbrella of many numbers of different compendiums of protein identification. So each year we put out a draft of what we call the status of the human proteome, in which we correct for a lot of the mistakes that a number of people have actually made in terms of their identifications, and this is done year by year. And so we're still doing that at the moment, and currently, you know, the identification of true proteins around about 70%, with another 16% that is ambiguous. We have our other components which we spoke about later in the program, so I'm going to touch on these, the SRM Atlas, and we also have our online resource such as PASL, which allows you to input your SRM data so you can store this as an online resource, and people can view that as well as the reviewers. And then we have a number of different other sources, and the ones I'm going to talk about today is this SWATH components, where we have libraries and where people can download these libraries for their own use. But more importantly, our new developments in statistical analysis tools of SWATH data and how that can help you in your research. And finally, we also have components in which we made significant inroads into cross-link analysis, and that will also be spoken about later in this program as well. So as part of developing unified transitions, we really understand that, you know, when we started this off in looking at selected reaction monitoring, the biggest component was actually trying to define accurately what are all the fragments of each peptide, how abundant are those fragments, and how we can use those in the selected reaction monitoring. And so we have efforts that we've done to do this at a complete proteome stage, and that will also be spoken about. But the advantages here is that once we have this data, that we automatically create these libraries for what we can do in this SWATH or data-independent analysis, where we can open up the peptide fragmentation profile so that we let in a number of different peptides of a mass range, fragment them all at the same time, and then get all of the fragments read out in this convoluted and quite busy quantitative profile. And so and at this point here, not only having the libraries we can interpret the data, but also having the tools to statistically validate those results as well. But that gives us these advantages of really becoming quantitative and creating a digital record of that approach. And so Sam Bader and my group was leading a lot of this effort, and where we can develop these databases and libraries, and he's developed that into an atlas now in which we can store that onto the web, and you can download those libraries. We have the website swathatlas.org, so you can do that download. There's also efforts underway which would incorporate tools in which you can interpret your own information using other open source tools such as OpenSWATH, and including the SWATH Atlas into commercial online computational efforts such as BaseSpace, which is an Amazon Web type component. But our efforts have really also developed into this notion of developing new tools for statistical validation, which we call the SWATH Prophet, and more recently we've just finished developing one we call SWATH PTM Prophet, or SWATH Prophet PTM. It depends on which day that we talk about it. But we also have other tools such as Disco that's coming along that's also going to allow you to do analysis without libraries at all, which is akin to one by Alexei Nevshinsky, which is DI Umpire. But what really happens with SWATH, and so you have this component here where if you look at the MS1 survey scan, you have a very busy chromatogram and spectrogram of a number of different peptides. If you select a individual mass range, you still see many, many different peptides that are isolated that component. But if you isolate that and then fragment all of those, you get this rather complex fragmentation profile. And you need to have the tools to enable to not only interpret this information, but also provide statistical tools to make sure that your interpretations are actually correct and not falsely produced and then propagated throughout your experimental results. This is the type of results that you do get. You get a lot of quantitative peaks that you look like very much like SRM profiles. But with SWATH, you immediately recognize there are a number of different peaks that are also coeluting at the same time with these fragment ions, and that those transitions actually come from different peptides that you're fragmenting at the same time. And so it becomes quite crucial that you've got tools that can separate your positive identifications from these other transitions that are occurring at the same time and not being misconstrued. But in the end, you're able to get these identifications by using your libraries against the fragmentation profiling and getting some good results. And you can use these to monitor cells across different growth stages and a number of different biological experiments. And so here we're looking at a housekeeping protein that is running at the same level at 3 different growth stages. And if you look at a dormancy regulator protein, we can see that once it gets to a stationary phase, that this protein goes up in its abundance in the cell. And you can take those types of results and start to create these profiles in which you can use them to classify certain proteins based on perturbations within the cell and understand how those changes occur. But the goal here is really to provide tools in which people can actually use these type of results. And so one of the tools that we developed was the SWATH Prophet, which gives you a probability score based on your analysis. But we also came across a component which we could use that information to now mine all of the information within the chromatograms and work out where there are modifications without any prior knowledge. So this is unlike what you're doing with shotgun analysis, where you would go ahead and say, I'm going to do a search and I'm going to search for a variable modification of, say, a phosphorylation and do that analysis. And if I want to go back, well, I want to do a search for, say, citronylation and then do that analysis. This is not what we're doing here. We're actually, I'll show you how we change that. And we don't do a pre-identification of any modification. We look at it as a mass difference and then go back and work out what that probability would be. Now, what we're actually doing is when we identify those peaks that I showed you in the SWATH profiles, is that we use tools that are very similar to SRM, which we look at the peak shape and coelution to make sure that the actual transitions that we're quantifying on are actually coming from the same peptide, not that they're coming from different peptides at different peak shapes and also slightly different retention times. But we've placed in there things like intensity correlations, utilizing the high resolution, and also trying to understand missing fragment ion peaks in these chromatograms. And then we can create probabilities using mixed model scores that we can get using our decoys, and then provide a separation between false positives and true positives, and ultimately end up with peak groups that we can understand. But in general with SWATH, you always end up with a number of peak groups that are not as nice as what you would expect, and you get a number of different fragments that are coeluting, and you've got to try and work out which of those fragments or transitions are actually coming from a different peptide, and try and work out what those transitions really mean. So in terms of interferences, we really want to understand which fragment ions are originating from the different peptides, and also determine which unexpected fragment ion intensities are really there. And that really comes across by looking at the scores that we are able to obtain. And then hopefully you can design libraries that can avoid those cases, but it's not always the case. The other thing is you want to do this automatically as well. The large volumes of data that's being created make you avoid trying to do any sort of interpretation unless you can do this automatically. And so we developed a number of scripts to be able to do this, in which we can do a number of adjustments, and then also allow us to modify the analysis and also do direct manual intervention at the same time. And it really is trying to detect two types of interference that we have in the profiles. And so this is what really occurs. And so if you have two peaks or two peptides that are very close eluting, and they have some transitions that are also shared between those two, what happens is we can't really determine which of those transitions come from which of the peptides and what that contribution is. And so we need to understand how to separate those as we go through. And so in this component here, we can see that there are two transitions that are exactly the same mass, but are co-eluting, but at slightly different retention time. And so they actually form this abundance, which is much higher. And so that's really what we're trying to detect here. And so if we look at our script, the way we do this, we can take out each one of these transitions one by one and recalculate what the probability would be without that transition, and then work out which one of those transitions was actually interfering, and in the end get to a better probability, and show that that one transition was really a component of two separate peptides, and then recalculate that component, and in the end get a better probability. But what that's also enabled us to do is to understand protein modifications. And it doesn't have to be just post-translational modifications. It could be any other modification that you're actually doing. And so we understand that protein translational modifications have a number of different importances in biology for sure. But where do we get this information from? There's only a certain amount of information that's currently stored, and this is mainly from prior knowledge. We understand that we can get a lot of this information from SwissProt or databases such as Unimod, but it doesn't include all of the information at all. There's a paper that was recently published that tried to calculate what was the experimentally observed post-translational modifications, and you can see by far the phosphorylation, since everyone seems to work on phosphorylation, is by far the most identified modification, but there are numbers of other different modifications that are also identified, but these are much smaller numbers than what you can get from the ones that everyone searches on. I think with Jenny talking this morning, citronellation is a rather small component here with very few numbers of identification. But if you look back at what was calculated as putative PTMs, by and far, the numbers of putative PTMs as compared to what has actually been observed is a huge dichotomy. The numbers that we're expecting here is in the 100,000 expected PTMs, whereas here, for each individual one, we're only scratching at about 10% or less, or even further. And so there should be those types of numbers, and also many others that we don't know of. And so the way that we're doing it at the moment with shotgun mass spectrometry, we really need to know in advance what these modifications really are, and we're limited to the amount of modifications that we can search against because of the expense and computational time that we have. And ways to get around that is we have either two-stage searches can help, and you can also mainly only favour detection of abundant peptides. Now with data-independent analysis, we have lots of detection actually being occurred, but again it requires prior knowledge through a library assay of a modified peptide, or some way of developing that fragmentation profile for the assay to actually work. And here is what I mean by say if we have a phosphorylated peptide, not only do we need to know what the unphosphorylated peptide fragmentation would be, but also the phosphorylated fraction would be, which would change the parent mass and also change the fragments of either side of the B and Y ions, depending on where that modification would be. And so with a new software we've tried to develop is it now steps beyond the spectral library, and then it identifies the peptides from low-ranking peak groups that were assigned to unmodified peptides that were originally searched. And so the steps here is that we identify the naked peptides, because these are generally, we have all the fragmentation profiles for those, and then go searching for the modified components that have the same mass differences for that unmodified peptide, and then recalculate through our efforts what the differences are in the modification and also the parent mass. And so in this way, there's no need for a modified peptide assay, and there's no need to anticipate these modifications. We can localize to a certain extent where that modification is, as long as we have the fragment ions around there, and we can infer the modification mass, but more importantly, we can do this in an automated fashion. And so we can also detect modifications that are usually due to sample handling, and also amino acid changes from non-standard SIPs or using proteogenomic data to try and defer that and identify that. But the important thing here is that it's not only post-translational modifications. A lot of people will think that the experiments that they're doing by taking reagents off the shelf that could have been there for 6 months to even 6 years or even longer, and expect those experiments to work by using those reagents, and they don't understand the modifications that are actually occurring to the proteome as they actually do that. And it's quite surprising the amount of changes that can occur. So this is the way that we have utilized this at the moment. So if we do a SWATH analysis, we have a spectral library which contains all of the fragmentation profiles for peptides that we're interested in. Take the sample, do our SWATH analysis. And then we take this software component and we extract the ion traces for desired modification mass ranges. And so here we have our unmodified peptide that we know what the identification is, because it matches the transitions that we have. And then we search up and down the retention time, and also up and down precursor windows, looking for low-ranking peak groups that match some of the ions that either differ by retention time or differ by mass as well. And then we can go and calculate what the probability would be by inferring a modification mass based on the difference of that precursor mass ion, and then also if we define some of the fragment ions and work out which ones are actually missing, and then recalculate based on the mass difference on those different fragment ions. And in the end, we can compute what a probability would be based on these new extract peak ion groups by utilizing that mass difference and seeing whether those transitions are actually there and recalculating those. And so some of these you see have exactly the same peak group shape, whereas others don't, and then others actually do, which depend on which of the modifications we're looking at. And when we finally do our computed probability, we can see that these ones that have identified with these different modifications have high probabilities, and the ones that have low probabilities usually define those peptides that didn't survive that test in terms of identification. And so we can go through and look at a number of different peak groups, both in charge states, and work out what these modifications really are quite easily in an automated fashion, and then work out exactly where their modification is. But sometimes if we don't have all of the fragment ions, we can only highlight where that fragmentation, where that modification would be in a certain region of the peptide. And then you'd have to infer either what that modification would be. It would be most probably a phosphorylation of serine rather than a proline, so you'd say it would be there, or you just have to leave it as a modification in that area. And then we can, what it does is make new library assays based on the updated precursor ion, then go searching across different experiments as well. And so here you can see this is what the original unmodified peptide assay would be with the parent mass and each of the fragment masses and what those fragment ions would be. And then it revises that information by taking that mass, imputing into the new precursor ion and also the different fragment ions to isolate that modification in that area. And so it re-extracts this information in an automated fashion as we go through, and then recalculates that back and gives you that positive peak group identification. But it gives you scores now, and it can also validate your results. And so in the end, what we have for our identification, we really must have first the identification of the unmodified peptide in the first place. We have the fragment ion peaks detected in the unmodified peak group, and between 1 and half the peaks not detected in the low-ranking peak group, we need to have this for the presence of the modification. We can't have too many missing peak groups, or it won't be identified. And then also the predicted intensities must be fairly well to this modified peptide as well. And so we take that information all into hand to create that identification. And so here's an example where, with these assays, we mainly have just the top 6 ions in this assay. And so if we scan across all of this, we don't have all of the fragment ions, and it at the moment, it can't contain too many fragment ions either. And so in the end, we can only detect modifications in 7 of this peptide's 15 amino acids using our analysis. So that's where the difficulty is, that you can't get accurate sight of the modification, but you can infer at least of where it is based on this approach. We're able to create decoys now on the fly, and then to calculate the probability based on the information that we have, and correct for modified peptides that have similar retention times in multiple samples and replicates. We have also the scores that we have. We have a number of different parameters that are used for the scores, both peak shape and parent ion peak shape that we use, but also these other scores as well that are used for the information. And then finally, we have other information as well that we can try and bring into it as well, as much as possible, getting known modification masses that we already have. And then on top of this, we use some of our tools that we have in the transprotein pipeline, such as iProfit. We have a number of these different parameters that we use of the having no sibling modified peptides involved, or other retention time normalizations as well, and also no additional peptides being brought into it as well. So these decoys then get very harshly penalized. And so in the end, what we're able to do is to take this information and infer a number of different chemical modifications as well, not only from post-translational modifications, but many other modifications, and looking at deletions of amino acids and so forth. But a lot of this stuff needs manual interpretation as well. Here's a result that we looked at a number of different synthetic peptides as well as peptides within human urine, and we detected a vast array of different modifications, not only our regular post-translational modifications, but things like oxidation of tryptophan, dehydrations of aspartic acid, and conversions of methionine to homoserine lactone, and so forth. And these modifications can be attributed to the synthesis of the peptides, but also native as well. And if we look at the estimated false discovery rate, we look at how many have we identified that were not in the library, and in large part we have over 65% of these that were not annotated at all in any library. I'll just go through a couple of brief examples of these. And so here we have a peptide that had a duplicated internal glycine that we're able to detect, and this was also verified using shotgun mass spectrometry, in which we had a full B series and Y series through this insertion here of a glycine. So we are able to detect these point mutations and also point insertions at the same time using our software. And these are things that would not normally be searched towards. Looking at localization of other modifications such as transamidation of piperidine. This is a result from synthesis. We can take these results and draw lines across our mass analysis and define how many of those peptides we actually see in our results, and separate a lot of the identifications based on this approach, and then store this in a component which we're building called the modification atlas. And at the same time, we can also look at just a number of our identifications with our scores, and then validate those both as SWATH analyses and also shotgun analysis. And you can see by and large our probability scores all fit with our ability to validate these, except for one here that has eluded us in terms of our probability, but was also validated as correct, and we're still trying to build the tools around that. And so out of the 13 of these were easily validated by both techniques. And then a number of these have different modifications that could easily be attributed to modifications that when there were multiple amino acids at the same time, but localized to the one that actually had that modification. And then just a few more examples here is where we show we have oxidation of methionine can easily be detected. This is the naked peptide, the low-ranking peak group in the SWATH analysis, and then recomputing that for the missing fragment ions and the parent, we actually get back the data that was actually stored in that result, and actually show that now we get a good match as we go through. So we pick up both those ions here, and then also N-terminal deletion as well, where we see this is the native peptide that we expected to see, the low-ranking peak group that has missing fragment ions that wouldn't be detected normally, and then by making that mass modification, we get back those transitions that was already stored in that data, so we actually get that true positive identification. We do this for both multiple charge peaks, either doubly charged or triply charged, looking at phosphorylation detection, and looking at the differences in fragmentation profile based on those charge states, can still pick up the low-ranking peak groups, and then recover that information once you use this type of software to go through. Also, when you're looking at native peptides within urine that's been modified to a homoserine and homoserine lactone, we initially thought that someone it was supposed to be a normal sample from in the lab, that someone had been near some signs of bromide, but that really wasn't the case. This is a known effect to happen in urine, and so we picked up this peptide here by recalculating those mass differences. But we can also get things like unknown modification. That's still an area that we're trying to understand and what these unknown modifications really are. So in the end, we come up with these issues here that really is a puzzle at the moment. You're getting presented with so much data on modifications, trying to understand all of this information. And we have multiple modifications being localized in nearby amino acids, so it becomes difficult to explore all of those combinations. And in the end, we can report localization sometimes precisely, but sometimes within groups. And then the modification may not be in a database, and we're still trying to understand how many of those are false positive. And so in summary, we have a number of different tools that we've made to help all this area, but getting into these new tools of validation of data that's coming through from modifications will really speed up the field, and we hope that this will spur people on to utilize that kind of stuff. So in the end, I just want to thank these people in my group. Large part of this was Andy Keller, but many people have contributed to this and also past members of the group as well. Thank you.
So now I'll cover this part. So what actually happens once you have the library and once you have the SWATH map? So how does the analysis work, and also what interesting things can you do with SWATH files that you cannot usually do? I'll first cover some of the analysis software as well as the general approach that they use. I then will go into SWATH-Prophet, how SWATH-Prophet does it, and some special feature to it, as well as how you can pursue unanticipated modifications using SWATH-Prophet. I'll say that. So some of the softwares that you'll find that can handle SWATH files are, for instance, PeakView and Oneomics. This is from SciEx. This is either the version you can install on your computer, or Oneomics is a cloud solution or essentially PeakView as a cloud solution. Obviously, SWATH-Prophet, which is the best one. OpenSWATH is from the Abersold Lab, is also open source, and you might want to try that one too. Spectronaut is from Biognosis. This is a company. If you want to use this software, you need to spike in these IoT peptides. If you don't have them, the software doesn't let you analyze the peptide, analyze the data. These peptides are, you don't know which ones, or I don't know which ones they are, so you have to buy it from them, and that's how they make the money. But I think it's also decent software. Another type of approach I haven't talked about and I won't talk about is an untargeted analysis. Or I'll cover it shortly in the next slide, but I won't go into it. And there are like 3 softwares that can help you do that sort of analysis. I'll also show you, how I'll go into detail of SWATH-Prophet. The other softwares work fairly similar, since they're all based on the same statistical tool that does the job. But there are also some differences. First, I'd like to just shortly cover how you can actually get some results without assays. So what you can do is, if you have an MS1, you can look for precursor ion in there. And then you can go at the same spot in the chromatogram in the next MS2 from that precursor, and look, do you find some unfragmented precursors there too? And if you do, you can get an elution peak for the precursor as well, so in MS1 as well as from the MS2. Additionally, you can go in that range of the chromatogram, which is very narrow, like about 30 seconds or so, and look which of the ions in there are actually coeluting. So you don't look for specific masses as you do with the assay, but you look for all the masses you find in there. And then you get some elution peaks for like a lot of fragment ions, and you look at how of them, like how is the coelution of these fragment ions, and do they match any how is the elution of these ions, and do they match a fragment ion of or can you generate a precursor or an MS2 spectrum or a pseudo MS2 spectrum that you can then search with, like, Sequest, Mascot, Comet, or the software you learned. And then the analysis is pretty much similar to or the same as with Shotgun. This is a way that works, and it works better than I thought it would, but it doesn't work as well as the one with assays. However, the benefit of that is that you don't have to have the assays. You don't have to have the assays beforehand. So if you have like a very precious sample and you want to acquire that, like you have one shot and only one shot, you might just acquire it in SWATH mode and take that route, and then hope that at one point you'll or try to get assays in some other way. But at least you can like get some analysis done with it. It's also something you can do on top of the extraction with the assays, trying to get something novel. The SWATH, I show you here a comparison between the Shotgun workflow, so the workflow you learned using the TPP, as well as how the analysis pipeline would look like if you do SWATH-MS in analysis in the TPP. So what you see here is like, okay, you have 3 samples. You first convert the raw data to an open format. You then do a database search with a protein sequence database. You then use peptide profit for the statistical evaluation, iProfit, protein profit, and you get your result. For SWATH-MS, you still convert the data to an open format, but then you use a spectral library and SWATH-Profit to analyze your data. And SWATH-Profit is both essentially the search engine as well as peptide profit together. It was also developed by the same guy who did peptide profit. So yeah. After that, and that's kind of unique for SWATH-Profit, is you can feed the result file from that directly into the other softwares from the TPP. So you can use iProfit. So if you have multiple injections, you can actually gain some results back by using iProfit. And iProfit has specific parameters that were developed only for SWATH. And you can feed that directly into protein profit and get the same result file back from there. I think the other softwares that I showed don't do the protein inference or don't solve the protein inference problem, so that might actually be something you want to try. As I showed you before, the way the analysis works is you acquire your data in the SWATH mode with the particular windows you want to set, and then you convert it to an open format, the SWATH file, and then you do a retention time alignment. So you pick the libraries, you pick some peptides in order to align the retention time from the library to the retention time of the SWATH run you just made. And then you go through like one assay at a time. So for each assay, you specify a retention time window. You don't extract it over the entire chromatogram. That would take too long. And then in that retention time window, what you do for each fragment ion, you extract the traces. Where do you see that? And here it's only shown for 3 fragment ions, but it's the same if you use 4 or 10 or whatever. So in that, for every spot at the retention on retention time scale, what you do is you look at the intensity of the fragment ion, and then once you have these peaks, you score them. In this case, how well do the 3 align? Wait, there's just one, so that's probably pretty bad. Second one, it's good. The peak, the elution peak looks good. The they have the same fragmentation pattern as what you would expect, the same mass shift, and so that one gets a high score. Here you only see 2, and 1 is kind of, the illusion is funny. The intensity, the relative intensity is kind of okay, but yeah, and so forth and so on. You do that for everything. You do the same thing for these decoy assays, and you hope that they look more like this peak rather than this peak. One thing I should mention, and if you do shotgun mass spectrometry, you have a spectrum, and you match that one spectrum to a peptide sequence. In SWATH, you do not have a spectrum. So it's possible that that one peak group gets matched to multiple peptides. However, it's very, very unlikely because, I mean, based from the assay library, you can check that none of the assays actually has overlapping masses. But in theory, none of the softwares actually corrects for that. So that concept of a spectrum and you match a peptide to the spectrum does not apply to the SWATH analysis. And that's also one of the reasons why it is so kind of slow, because you have to extract each assay from the same window. And so it also takes some time. But it works. So yeah. In case of SWATH-Prophet, what you get is actually accurate probabilities. So it's very similar. The result file that you get can be seen very similar to the result file you will get from Peptide Atlas. Peptide-Prophet. So in this case, what we've done is, and so this is work, I should probably say that, I've done with together with Andy Keller. And in this case, what we've done is we looked at peptides that, or we had synthetic peptides and just analyzed them in the mass spec. So we did know which peptides are actually in there. And then we extracted that with a decoy, a set of decoys with the scrambling and everything. But we also used assays of peptides that were not in there. And so this is like, on one hand, you have assays that do not exist, or from peptides that do not exist. And on the other hand, you have assays of peptides that are valid, but they are not in there. They should not be there. And so you can compare the score profiles of the two, which should be this large peak of the negative distribution that you always seen before, or that Luis showed you yesterday. And you just look how well do they overlap. And so they overlap fairly nice, or fairly good. And so you can say the computed probability are actually accurate. And this here is like just to show you like it's very close to the diagonal, and the error bars are acceptable. Additionally, I've already said that, but again, SWATH-Prophet can use or take use of iProfit. So you have replicate injection, replicate SWATH injections. You might get a boost, and this data set was generated by a dilution series of these peptides. And so this boost mainly came from the low intensity peptides. So something interesting you might want to do is, if you have a proteins you're interested in, you can have one run in which you know these peptides show up. So say you order the synthetic peptides in the light version, and you spike them in the library, so you do know these peptides are there. You analyze that, and this is a run. You know these peptides are there. Now you have your sample, and there you're not sure if they're there, but because you can use iProfit, you have a better chance in finding them, because this run with the synthetic peptides helps you boost the IDs, unlike the specific IDs, because it requires essentially that the IDs are at the same location. I'm going to show you that on Monday in real life. But this here is Andy Keller. He did the SWATH-Prophet. Yeah, they use the same scoring system. They use some of the scoring is similar to the one from SRM. So it does use peak shape and coelution, which is like depicted here. However, it can use some additional scores like it has high resolution M over Z, and it doesn't look only at one mass, but it has the full MS2 spectrum. So it can actually look at the isotopic distribution of the fragment ion, which is something you usually don't have. And you can also look at fragment ion, like additional fragment ions. So fragment ions you don't have in the first pass of your assay if you extract that. You can go back and re-extract a new one, or you can look at these. It does that at the apex. Some challenges with the data analysis. I mean, peak groups should look like that, but they normally don't. This is the things you see, and this is the things you get. I mean, not that bad. Two of the bad scenarios are like if you get interferences, which is like if two peptides have a common fragment ion and they may not coelute perfectly, but they overlap in the coelution, then you get things like that. We call that interference. Or you get a fragment ion that is missing. And the fragment ion might be missing because it's just too low intensity and you're not seeing it. Or it's also possible with what I've already shown you with the oxidized methionines, it might be modified, and that fragment ion might actually cover the modification, and that's the reason why it's not there. And so I like to show you how SWATH-Prophet can actually handle that. In case of the fragment ion, of the interferences of fragment ions, there are two kinds, or for the analysis, there are two kinds that one has to distinguish. One is if your assay library contains two assays of which you know they kind of coelute, the retention time, or the estimated retention time is very close to each other, and they have fragment ions that are within the mass range that you extract. So you're not extracting with like, this is the mass period. You always have like a slight mass error that you allow. And so as shown here, these are two peptides, two assays of which you know from the library. That's why we call it intra-library. Two peak groups that overlap. And you see, actually you should have here the B ion, the fragment ion of this peptide called B, for some reason, that should elute here, and you have the A. Yes. So fragment ion A and Y, they have the same mass. Yes, they do. Down here is listed. And so you only see one peak like this. But you would like to see a peak like that and a peak like this. In case you have the library, you can just browse through and you find these instances. And in case you find like a result, if you find that assay has a hit, you already know that, hey, you have to be careful with A. It might have an interference from this Y ion, or this ion named Y. Separate scenario is if you have a peak group from an assay in your library, but the interference does not come from another known assay, but it just comes from your sample. It's just in there. And in that case, again, it looks similar. You have A, C, and D that nicely coelute, and you have the good scores, but the trace of the ion B here, the shape doesn't look right. What you can do in that scenario is you can remove each of the fragment ions and recompute some of the scores that are affected by this, by the not so perfect elution. For instance, the observed intensity, because you have an additional peptide there, the intensity might be off even at the apex. Like here, for instance, here the Y one, that one is clearly off. It's too high. So that score should be not very good. The peak shape, obviously, because you have like two elution peaks that overlap, as well as the peak apex time. it might be the same, but or yeah, it might be off. So what you can do is you just remove each of these fragment ions. So in the first case, you remove A, you recompute it, and you see, okay, before it was 0.9. If you remove A, it's 0.73. It's worse. We don't like that. If you remove B, you recompute all the scores. You get 0.98. That's good. And you do the same for C and for D, and you see it's still worse. So if you remove B, you're better off. So that one is very likely to have an interference. What you also can see is that the intensity correlation specifically increases. So you go from a 0.74, which is like borderline, it's not that great, to like 0.82, which is like, it's not perfect, but it's like good. The intensity, it's a normal correlation. So 0 is it doesn't correlate at all. 1 is you got it perfect, and -1 is you got it the wrong way around. So, but yeah. And then what the software does in this case, you do have relative intensities for both assays. So what you can do is you can use the other intensities for both of the peptides to estimate the intensity that you should have for each of these fragment ions, and like this separate the intensity. In this case, you don't have that, and you only have it. You can only do it for one, and then whatever remains just belongs to like some ion in your sample. In that case, you won't find out what it is, but usually you're happy just having the interference removed. Here are some of the these interferences that you can see, and what you show you is these are synthetic peptides, and they are either neat, so that means in like just buffer. There you don't have a background, and these ones here were in urine background, so that peak here comes from urine and interferes with it. Here's the same thing with the purple line, an interference from urine that is gone in the neat solution. And you see this one here is supposed to be the top fragment ion, and that one is reduced, I think down here even. What we can do is we can calculate an interference strength, and that is simply like what fraction of that intensity, no, what fraction of the total intensity actually comes from your ion, and how much is like from the interfering species. And so what you can see is that there are between the two, the score base is very good at identifying like very large interference peptides. So these ones here are large ones. The ion that you look at, the ion that you're interested in, only makes up a tiny portion of, or in this case 10, in this case 20% of the total peak. In that case, we compared this interference strength is also computed accurately. What we could do, because we had the synthetic peptides both in neat as well as in with urine background, we did know what the actual area under the curve is supposed to be. So like in this case here, we knew that it should be that much and not this much. So we could compute the actual area, and we knew like how much is the interference strength and how much was computed. And so you see this is a fairly straight line, and only for a few of them you get some deviation, but you can look at that. This is interesting because it also shows that only some of them have so you see there's a decent fraction of peptides that actually have an interference, and it's actually worth doing. Although I have to say in this case we only look at peptides that are identified anyway. So what we additionally can do is look for modified peptides. So look for peptides that are modified, but you don't know which modification it is, nor do you know which site it modifies or which amino acid it can modify. And the way we can do this is, assume you have that peptide, and the peptide gets identified nicely. This is the peak group, great. I showed you before that for each of these assays during the extraction, you actually get multiple peak groups that you score, and you only pick the top one from the scores. If you have a peak group like this, that gets a lower score, but for these 3 fragment ions, you still get a very decent score, like the scores perform very, very good. There's only 2 of these are missing. What you can do now is you can pursue these and see like, hey, what went wrong there? And in that case, you can see, okay, the 3 that you see in both cases, they go until here. So that part of the peptide is probably fine. These 2 are missing. So if you have some mass shift at either the S or the P, you won't see them. So your hypothesis is that these are modified. If they're modified, you should identify fragment ions that correspond either to a fragmentation to the Y8 or the Y10 that has the same mass. You can also look at the precursor ion, and if you have the precursor ion that shows the same, or if you see an ion at an intensity at the same mass shift, you can get fairly certain that it could be a modification. So what you do is at the apex time, so here, you take the MS2 spectrum as it is acquired, and you make a so-called modification mass spectrum. That is, for every, you look at the intensities you see in respect to the mass of a Y8 or a Y10. So in this case, if here it would be 0, this is the mass, the m over z charge of the Y, that's Y9, of Y9, and then you see, okay, you have very close by 2 intensities, then it goes a while, then there's another one, and so forth and so on. And if you align these mass, these modification mass spectra all underneath each other, you should find one peak that is present in all of them. And that mass peak should not only be there, but it also should have the intensity at the isotope profile. That should also be there. So you know that if you find a mass here at that location in the modification mass spectrum, at +1, you should have 1 too. At +2, you should have 1 too. This is, the isotope profile. In this case, you have +80. So now you know that if you add +80 to either the serine or the proline, you could explain, or you could explain that peptide that eludes here by saying either the serine or the proline is modified with whatever makes up mass 80. What could that be? And so in that case, what you do is you can't say that with that assay. So you need to go further, and you need to take the Y8 fragment ion. The Y8 fragment ion is not in the library, but that doesn't matter because in this spectrum, in the apex MS2 spectrum, you should still see a mass at either the one that is modified, and then if it's modified, then the proline has a mass of +80, and if it's not modified, then the serine is most likely modified. And in that case, you find, so here you see, okay, if the Y ion is at 0, at 0 in the modification mass spectrum, it's at the serine, and if it's at +80, it's the proline. In this scenario, you do probably know what the mass shift of 80 at the serine could possibly be, but it's also possible that you get here a mass shift, and you cannot explain that. So this is the tricky part of the whole this analysis. The way it works, I mean, with with the example I showed with the modified methionine, the oxidized methionines earlier, you can think of how that might work if you use the same precursor window. However, we managed to get that running with like across windows and even across samples. So what you can have is you can do a phosphor enrichment. You can dephosphorylate everything, acquire that. There you identify your unmodified peptides, and then you can acquire the phosphor enriched sample and look in there and find phosphopeptides in there. That works. So it doesn't, that peak group here does not have to be in the same run as that run here, and obviously not in the same window. So the way the analysis would work is you get your spectral library, you get your sample, you acquire your data, and then of the identified peak groups, what you do is you look in the same window as well as different windows for like slightly imperfect peak groups. So some that miss these fragment ions, and with a particular shift in retention time. The shift in retention time might be helpful because you know, say, for instance, that oxidized methionines, you know, they're always earlier. Yes, they're always earlier. I forgot. And so with SWATH-Prophet, we call it SWATH-Prophet PTM because it has this post-translational modification capability. What it can do then is it can look either like kind of a focused mode, where you know, okay, the sequence that it can be localized contains a methionine and it's plus 16 and has a delta retention time in a particular range. And we can be fairly certain this is an oxidized methionine. But you might also have cases like this where you see, okay, minus 56, it's either asparagine or glutamate. Yeah, -1 in the middle of the sequence, but the coelution does not look that great. So yeah, that's probably wrong, and probably most of them are in this category. Or you might get in a different window even larger modifications, such as phosphorylation. This one here, I have to see. Yeah, so this is an example that we have. We analyzed, or we validated that approach using the dirty peptides, or the synthetic peptides that we had. The reason for that is, if you order peptides, the synthesis conditions are so harsh that you get all sorts of modification. And at least neither me nor Andy knew what kind of modifications to expect. And there's a whole range of them. So we thought that's a good idea to do. Although for biologists, this is not really interesting. But we could show that, yeah, for the oxidized methionines, it works pretty nice. So if you have here the real synthetic peptide, here you have one that has the largest fragment ion missing, as well as the dashed line here is the precursor ion. And then, one of the lower ones too. So Y7, Y10, and the parent ion are gone. And so if you look for possible modifications, you can explain that with a modified, an oxidized methionine, and you get a nice peak group back. In this case here, the top rank peak group looks nice. The next one has like just the top fragment there, the most intense fragment ion, but the precursor is gone, and like some of the other ones, 3 of them, yeah, like the B ions. And so what in that case happened, you have -129 from this range. So that's probably deletion of the N-terminal glutamate. So the kind of modifications that we found were like not in the synthetic peptides, as well as the urine background. We could also analyze that one, obviously. We found some that are expected just from sample handling, and like if the sample gets old, that's when like you see deamidation or oxidation of methionines, as well as pyroglutamate. You can identify them. So in general, our approach works, and our approach works to the extent that it can be anything. It can be like, yeah, you can chop off the N-terminus, you cannot chop off or add to the C-terminus. You could have an internal amino acid addition. So it can be that there is a peptide in your proteome that only has one amino acid just in the middle inserted, but otherwise it's the same. And so like this, you can identify that. I'm going to show you a result from the synthetic peptide where that actually happened. You can also, this one here, even the company that made the peptides didn't know what that was. That was a piperidine transamidation of aspartic acid. But yeah, so we can identify pretty much any sort of modification as long as it's like if the precursor doesn't go above 1200 or 1250. We then went and so what we did with the synthetic peptides is we had about 1200 of these pooled together and analyzed. And so if you have here a deamidation of a particular peptide, you know, or we knew that must have happened in the tube where the peptide is. An alternative would be that it's just like, well, it's a lucky coincidence that you see these fragment ions coeluting there, and it's actually not that peptide that is deamidated, but just some background. So what we could do is we can go take only that one peptide and analyze that in SWATH mode, and we should see that the proper way and the deaminated way, or we should see it with and without, with the oxidation on the methionine and the reduced way, and so forth and so on. And so we did this analysis for 14 of these that we could not explain already with assays in the assay library. So some of the usual, these ones here you usually expect, the deamidation, the oxidation, and the pyroglutamate. Some of them we already had as assays in the assay library and could explain like this. For these 14, however, what we did is we took the peptide, or yeah, Uli went to the freezer and had to dig through all of the peptides. And we then looked for specifically, do we see it unmodified and with the particular modification? And we could validate all but one, but this one had a very low probability. So in this case, it might be just a false positive. The interesting case is the red one here. And this had a sequence where there are two glycine, or at the N-terminus, so from the tryptophan till the alanine, we could say, or Andy could say, there's a plus 57. And whoever does mass spectrometry knows plus 57. Well, this is if you use iodocetamide. That's what's attached to your cysteine. So he said that's from the alkylation. And I said these are synthetic peptides. They don't touch iodocetamide. Until we figured out that 57 actually matches also the mass of glycine, and that it could be that these two glycines actually happen to be three glycines. And during the synthesis, like in one case, for instance, the blocking didn't work very well, and they got two glycines added. And sure enough, if we did a shotgun run and added this sequence to the database, we could see a very nice fragmentation spectrum covering exactly that spot with the three glycines. And this also shows you one of the troubles you have if you do that analysis. You only have a delta mass. So in that case, you couldn't say, so we don't have any evidence if it's like an alkylation, oh yeah, without that spectrum. We don't know if it's alkylation or a glycine. And the same thing might happen if you have some other nice modification. For instance, trimethylation and acetylation have a pretty close mass too. So there you might not know which one it is.